Autophagy is one of life's most elegant survival tricks, a process that allows a cell to clean house and renew itself. Imagine a miniature recycling plant operating inside every cell, dismantling worn-out parts and feeding the raw materials back into the system. Without this quiet cycle of renewal, cells would drown in their own waste and lose the ability to adapt to stress. At the heart of autophagy lies a choreography of membranes. It begins with the birth of a thin, crescent-shaped structure called the phagophore. Like a net being cast into the cytoplasm, it expands to capture unwanted cargo, damaged proteins, old organelles, even defective mitochondria in a process known as mitophagy. Once the phagophore closes, it forms a sealed chamber called the autophagosome. This double-layered bubble then fuses with a lysosome, the cell's acidic chamber of enzymes where the contents are broken down into reusable building blocks. Unlike the blunt force of bulk degradation, this is precision recycling, capable of selectively targeting components to maintain balance and health. The decision of when to initiate autophagy rests on molecular sentinels that sense the cell's energy and nutrient state. Chief among them is the mTOR complex 1, mTORS1. In times of plenty, mTORS1 keeps autophagy silent, ensuring that cells focus on growth. But when nutrients run scarce, its repression lifts, freeing ULK1 to trigger the cascade that builds autophagosomes. Another sentinel, AMPK, listens for shifts in energy charge. A drop in ATP, the cell's fuel, pushes AMPK into action, tipping the balance toward survival through autophagy. Transcription factor EB, TFEB, adds another layer of control, slipping into the nucleus during starvation to turn on genes that build the autophagy and lysosomal machinery. This machinery is not merely a background process. It is essential during nutrient starvation, in aging cells where damaged components accumulate, and in defense against pathogens. Autophagy can act with surgical precision, removing only what threatens the cell's equilibrium. In this way, it is more than recycling. It is a guardian of cellular identity, ensuring that cells can endure stress, preserve energy, and maintain their place in the living fabric of tissues.